Welcome to this explainer. Today, we're diving straight into the wildly fascinating world of assisted reproductive technologies in animals. You know, for thousands of years, humans have selectively bred animals just by choosing the best of the herd. But over the last century, we've developed systematic, highly advanced techniques to literally bypass natural mating altogether. We're going to explore the absolute cutting edge of veterinary science, moving from the very first embryo transfer experiments all the way up to modern cloning, intense lab manipulations, and extreme biosecurity. It's a crazy journey, so let's get into it.
Here's a quick look at our roadmap for today. We'll start with the major milestones in animal art, then break down the actual embryo transfer process. After that, we'll dive into the in vitro revolution, look at how we evaluate and preserve embryos, and finally, wrap up with some of the extreme manipulations and biosecurity protocols.
Section 1. Milestones in Animal Art. A History of Innovation. Let's set the stage with a bit of history that makes all this incredible science possible.
It is actually wild to think about how incredibly fast this field has moved. We went from a basic embryo transfer in a rabbit in 1890, carried out by a scientist named Heap, to fully cloning a mammal in just over a century. By 1951, we had the very first calf born from an embryo transfer. This tech even crossed over into human medicine in 1978 with the first IVF baby. And then, of course, boom, 1996, the world meets Dolly the sheep, the very first mammal cloned via somatic cell nuclear transfer. Talk about a massive leap in a short time.
To really grasp why this technology is such a game changer, imagine a single, genetically elite female cow having 15 calves, all at the exact same time. Through a program called multiple ovulation and embryo transfer, or MOT, breeders use superovulation to collect multiple embryos and transfer them to surrogate mothers. This allows us to radically speed up the genetic progress of an entire herd, basically creating large, full-sibling families in a fraction of the time nature would normally allow.
And if you're thinking this is just some niche experimental science, think again. To put the sheer scale of this into perspective, in 2015 alone, more than 520,000 naturally derived bovine embryos were transferred globally. So we're talking about a massive, standardized procedure that is literally driving global animal husbandry today.
Section 2. The Embryo Transfer Process, from donor to recipient. Let's look under the hood and see exactly how veterinarians move a developing embryo from one animal to another.
The whole shebang starts with something absolutely crucial called superovulation. This is a total prerequisite for animals like cattle or horses that naturally only release one egg at a time. By using highly specific hormonal treatments like FSH, vets can actually stimulate the donor's ovaries to produce multiple mature eggs at once. And the incredible part? They do this while completely maintaining the normal physiological health of the animal.
Pulling this off requires a strict four-step sequence. First up, you superovulate the donor, but here is the real trick. You absolutely have to synchronize the estrous cycle of your recipient animal at the exact same time. Second, you artificially inseminate the donor. Third comes the embryo collection, which is usually called flushing. And finally, the transfer itself. That synchronization I mentioned, it is make or break. The recipient's reproductive environment has to be perfectly ready, meaning she needs to be within plus or minus 24 hours of the donor's cycle stage, or the pregnancy just won't take hold.
Now there's actually a massive contrast in how vets pull these embryos out, depending on the animal. For cattle, it's pretty straightforward. They can use a specialized catheter and fluid to gently, non-surgically wash the embryos right out of the uterus. But smaller ruminants like sheep and goats, that's a different story. Getting a catheter through their cervix is super difficult, so vets often have to use precision laparoscopic surgery or even general anesthesia to retrieve the embryos directly from the uterine horn.
Section 3. The in vitro revolution. Life in the lab. This is where things get truly sci-fi, as we take the reproductive process entirely out of the animal and into a petri dish.
Using something called ultrasound-guided ovum pickup, or OPU, vets can repeatedly retrieve immature eggs directly from the ovarian follicles of a living, elite female. This is huge because it lets us completely bypass traditional infertility issues like blocked oviducts. But get this, if a highly valuable animal dies unexpectedly, veterinarians can surgically remove the ovaries and extract those eggs up to 5 hours post-mortem. It's an incredible way to save a crucial genetic line. After that, everything, maturation, fertilization, and culturing happens completely in vitro, right there in the lab.
Section 4. Evaluating and preserving embryos. Quality control. Once we've engineered these embryos, how do we actually know they're healthy enough to survive being transferred?
You might be wondering, do lab-grown embryos look exactly the same as natural ones? Well, it turns out human intervention has some real physiological consequences. Under a microscope, in vitro embryos look fundamentally different from natural in vivo ones. They're noticeably darker, they carry a much higher lipid or fat content, and their cellular structure just isn't as tightly compacted. Because the artificial culture environment changes how they develop, veterinarians actually have to use totally different criteria when visually grading them for viability.
And because simple visual checks aren't always enough for these lab-grown embryos, scientists have had to bring in some incredibly advanced tools. We're talking multi-photon laser scanning microscopy with fluorescent markers to check cell structures. They also use PCR gene expression analysis. That allows them to not only check if the right genes are firing, but to actually determine the sex and fully genotype the embryo before it ever gets transferred. They can even sample the spent culture medium, basically testing the fluid the embryo is growing in to measure its metabolism and predict if it'll survive.
If an embryo passes all those rigorous tests, it doesn't even need to be transferred right away. By using specialized cell freezers, we can slowly step down the temperature and store the embryo in liquid nitrogen. We are effectively hitting the pause button on time itself. This is absolutely massive for the industry because it means you can ship elite genetics anywhere in the world in a small, thermos-sized tank, completely avoiding the nightmare of transporting a live, 1,000-pound animal.
Section 5. Manipulations and Biosecurity, the Extremes. Finally, let's explore the absolute edges of this science, where biology, engineering, and global security all collide.
At the very extreme end, we have technologies like cloning via somatic cell nuclear transfer. This lets us practically copy elite genetics or even create highly specific transgenic disease models for research. But nature doesn't always love being engineered. Constantly moving gametes from dish to dish, exposing them to artificial light and temperature, it all comes at a biological cost. Highly manipulated embryos suffer from much higher rates of pregnancy loss, and sometimes they develop what's known as abnormal offspring syndrome, where the artificial handling actually messes with epigenetic programming, leading to unusually large birth weights or congenital issues.
So, I want to leave you with this final, provocative thought. As we continue to push the boundaries of creating life in a dish, we are constantly walking a tightrope. On one hand, we have very real physiological risks, like abnormal offspring syndrome when we handle gametes unphysiologically. But on the flip side, moving livestock globally at the embryo stage rather than shipping live animals drastically reduces the spread of global diseases and massively improves animal welfare. It's an incredible, high-stakes balancing act for modern veterinary science. So where do we draw the line? It's definitely something to think about. Thanks so much for joining me on this explainer.